Okay, so this is the fourth topic from Unit 2 of the Higher Human Biology course, which covers antenatal and postnatal screening. Now, to start with, to just explain what these two words mean, anytime you see the prefix ante, it means before, and post more commonly means after. So antenatal means before, or basically before birth. So throughout pregnancy, antenatal screening can go on, and then after birth, postnatal screening will occur. So we're going to concentrate on a couple of different aspects of antenatal screening, and focus on one idea, one sort of thing that's looked at after birth, although there are loads of things, but that's one of the tests that's done specifically. And then once we've done that, we're going to move on to look at some work that is done on family trees in the course. So this idea that quite an awful lot of all genetically inherited conditions will follow some pattern within a family tree. And what that means is that as long as we understand what kind of disease or what kind of condition it is and how it's inherited through a family tree, we can start to make predictions about offspring before they're even conceived. So you can start to think, well, if I know enough about my parents and my grandparents and those conditions, and the person I'm about to have a baby with also knows that information, then we can have a pretty good idea as to the chances of our offspring having a condition or not having a condition. So we're going to talk about that. I'll rub the board off and we'll talk about that part of it. Okay, so firstly we're just going to concentrate on the kind of four titles that are on the screen just now. Firstly, ultrasound scans. What you need to know for the course is ultrasound scans, of course, are a process that we can actually look at what's going on in the womb during pregnancy. So that's obviously, as it's colour-coded red, it's an antenatal screening method. The typical pregnancy will consist of two scans. First of all, a dating scan, and secondly, an anomaly scan. Right? Now, the dating scan, quite simply, is pretty well named for a start. It allows you to work out the stage of the pregnancy and, in turn, the due date for the baby. That will take place between 8 to 14 weeks. At that point, they do a wee measurements on the baby's head based on and try and work out the age of the baby and how it's developing in the womb. So that's the dating scan, and that's what that's for. The anomaly scan is done a little bit later than that, between 18 to 20 weeks, and that helps to look for potential issues, physical abnormalities that might be more obvious by that point. So you can start to get an idea for if your baby might have any sort of, well, not any, but some forms of condition that can be detected by anomaly scans. So the dating scan's first, anomaly scan is next. That's all you really need to know about that. Just make sure you understand that you've got these brackets for the number of weeks that they take place in, and roughly what they're for. Blood and urine tests will basically happen, and it's not the baby's blood and urine, it's the mum's, obviously. So the pregnant woman will give a blood or urine or both sample pretty much every time that she visits the hospital, and they'll be doing that test for all sorts of marker proteins. So tests for marker proteins. And marker proteins can tell you all sorts of things. They can indicate things about the baby's health, but they will also indicate things about the mother's health, which is very important to keep a track of throughout the pregnancy. One thing that's worth mentioning about blood and urine tests is that if the test's taken at the wrong time, they can give false positives. False positives, and all that means is that you might get a result that says, oh, there's something wrong, when actually there isn't. So you've got to be aware of the time that these tests are taken, but they are fairly standard as part of the whole process of antenatal screening. Diagnostic testing can be done as well. Now, basically there are two different types of diagnostic tests, but ultimately they both result in the same thing, and that is that we produce a karyotype. A karyotype is for want of a better expression, it's a photograph of all of the pairs of chromosomes. So you would know from your basic knowledge of human genetics that there would be 23 pairs, and you can look at 23 pairs all lined up together. What they can do with the computer now is they can take an image of the nucleus of a cell, which will no doubt be dividing, because most cells in a fetus are dividing, so they'll all have reached this little like wound up stage where all the little chromosomes are visible, and then they can use a computer program to find the pairs and arrange them in 23 pairs. I'm not going to draw all 23, but the idea is a karyotype would be all 23 pairs. One of the things you can detect is if it's a female, you would have 23 pairs that would all be exactly the same size. If it's going to be a boy that you're having, you would look at the karyotype and you would actually see in the 23rd pair there would be a big X chromosome and a wee small Y chromosome, and that would tell you that the baby that's developing is going to be a male. So anyway, how we get the karyotype is we need a sample of the cells from the baby, right? So the way that we do that, there's two different ways to get the cell in order to obtain the karyotype. Okay, so the idea is that both of these tests that I'm going to tell you about in a little second result in you having a cell which you can look at under a very powerful microscope, and using a computer program can pair off all the chromosomes. It would be a check for like Down syndrome as well, where that involves having an extra chromosome. So you would be able to tell if the baby was going to have Down syndrome, because it would have 47 chromosomes rather than just the 46. So I'm just going to get rid of this sort of drawing of the karyotype to try and talk about the two different kinds that you get. So there are two different types of diagnostic test. The first one is called an amniocentesis. Amniocentesis. So amniocentesis comes from the word amniotic. The amniotic fluid is the fluid that sits in the womb and surrounds the baby. It acts as a sort of a shock absorber largely, but what you will also find it's filled with is lots of little cells that have flaked off the baby. So the baby's cast off these cells which will be floating about in the amniotic fluid. So all you do is, it sounds quite nasty actually, but they take a needle and it goes right into the womb and they withdraw a little bit of amniotic fluid. Now you wouldn't do an amniocentesis on everybody because it is, it's not particularly nice. You would need to have a reason for it, and probably an anomaly scan showing something up would lead you to think, well, let's have an amniocentesis, let's just double check everything's okay genetically, at least what we can tell from the karyotype anyway. But what amniocentesis, as I said, is you take a little bit of the amniotic fluid from around the baby, and that is then looked at under a microscope because there will be baby cells in that. The second way of obtaining a karyotype is something called CVS, chorionic villus sampling, and that is, CVS is absolutely fine, incidentally, you don't need to know chorionic villus sampling, but what this is you're taking cells from the placenta, some of the baby cells from the placenta. That is a much more invasive technique. Chorionic villus sampling can be done earlier in pregnancy, so there's an advantage of knowing the information, getting a karyotype earlier, but the downside of it is that there's a higher risk of miscarriage if you have a CVS taken. So if this one can be done earlier in pregnancy but has a higher risk of miscarriage, this one here, an amniocentesis, has to be done later, so you don't get the information. Once you've got the information about potential problems with the baby, it's closer to the birth. So that's the disadvantage of amniocentesis, but it's far less invasive and less likely to result in a miscarriage. You have to understand the pros and cons of amniocentesis versus CVS as part of the course. That covers everything in terms of antenatal screening. We're now going to focus just very briefly on postnatal screening. This is the idea that we start to test things about the baby after it's born, right? So you do things like a hearing test, there's other tests where you You might see how the baby reacts to not being dropped, but being sort of like leant back quite quickly. You'll see the baby will have a fall reaction. There's certain things that you're looking for the baby to have. But another thing that all babies are tested for is a condition called phenylketonuria, PKU. Now, PKU is a genetically inherited condition where a person cannot break down an amino acid called phenylalanine. Now, phenylalanine is quite a common amino acid found in the diet. Very specifically, it's found in artificial sweeteners. So things like Diet Coke and diet drinks can be really, really bad for you. Besides gas bagging you, they can actually contain sources of phenylalanine. Now, phenylalanine is this amino acid which the body can't break down. People that don't have PKU will break it down into something called tyrosine quite quickly. So phenylalanine is broken down into tyrosine, and that's in ordinary people who don't have PKU. If you do have PKU, because that doesn't happen, because it's a fault with the enzyme that breaks phenylalanine down into tyrosine. We have mentioned this actually in the metabolic pathways topic as part of Unit 1. But if you've got PKU, you have to be tested very early on, and if you do have PKU, you'd be very careful not to give any source of phenylalanine to a PKU sufferer, because as a child, as a toddler, or a baby developing into a toddler, you can get serious damage done, especially to the brain and the spinal cord, with this phenylalanine, because it builds up in the body because there's no way to break it down. And it's a legal requirement. You'll see this on anything that's sugar-free. Chewing gum is quite a common one, where it will say in very, not big letters because it's a small packet, but in big as letters as you can really on the back, it will say contains a source of phenylalanine. So really important that you understand that having PKU means you can't break down phenylalanine. So that is a summary of the ante and postnatal screening that can go on. At this very moment, I'm looking at this running time of 12 minutes, and I'm thinking I'm going to break this up into 2. So this is the first of 2 videos, where the next one I'm going to look at family trees. I'll rub the board and I'll start on family trees. So as I said, that's a decent summary of what I've covered. Just watch it several times, make sure you take in everything about ultrasound scans, blood and urine tests, diagnostic testing as antenatal processes, and finally the idea about PKU being looked for. You just take a blood test, you look for a certain protein, and that's the one prime example of postnatal screening that you need to understand about. So this is one of 2 videos which will help you with this topic, which will stop now and we'll move on to the next one.